First of all, I'd like to thank the organisers for the opportunity for our group to present how we're addressing some of the challenges. And the two challenges I'm going to talk about are reduced time to market and then touch a little on advanced manufacturing. So I think this follows nicely from Vivek's talk, and anyone who's involved in drug manufacture, drug product or drug substance will have seen this idea of the ICH Q8 pharmaceutical development, that you decide what do you want to manufacture. Is it sterile? Is it oral? Design, that's the basis of your design. Designing quantitatively what properties that will have, and then coming up with the materials and the process to manufacture it, and then your control strategy for manufacturing. So that's kind of a simplistic overview of it. And why I picked this topic, and I thought it's interesting, especially when we have lots of researchers who are back where I was maybe 20 years ago. And I did my PhD on solid amorphous dispersions, and I was in a company making tablets, and we were working with penetration enhancers for oral delivery. And the penetration enhancer powder didn't flow well. It didn't flow and it didn't compact. So what do you do? If you're in solid oral dosed form, you make a granulate. And we made a wet granulate, and because it was quite a surfactant material, it made a really hard granulate. So normally with a granulate, it's very diverse in particle size, so you mill it to get a kind of a uniform particle size. And our granulate wouldn't go through the mill. So there was a few of us working on it, and someone said, do you know what we'll do? We'll put it in a bag and we'll hammer it. And it worked, and it tableted, and we thought, happy days. So we went to our boss, and our boss said, well, how are you going to scale that up? And you can't get big hammers in pharmaceutical plants. So you could say, well, why don't we get a more aggressive mill? But the issue with getting a more aggressive mill is the idea of granulation is to bring your powder particles together so they'll be bigger and they'll flow. So putting it through a fluidizing mill or a more aggressive mill will bring you back to the powder that you started with. So I suppose the point to this is that it really brought home to me, when we're designing formulations, we need to be designing them for manufacture, because all drug products, the objective is that they're going to be used by patients, so they're going to be products, and they'll need to be, in most cases, manufactured at scale. The other thing kind of to highlight this is, if we look at the recent development of the COVID vaccines and some of the scalability issues related to those, kind of highlighted these issues. So when we think about a product, we can think about it as the materials, scaling up the equipment, and scaling up the process. So if we look at the genetic, so that was our Pfizer and our Moderna vaccines, and that was a really new technology. A lot of the raw material issues, we saw highlights around those, but they managed what was very interesting. The microfluidics, or the lipid assembly, was seen as the biggest challenge, but that was kind of overcome. I think there was a lot of learning from other gene therapy products in the area, and we got eventually to mass production. There was also issues around the viral vector vaccines, and I think Paula touched on it nicely yesterday when she was kind of talking about cellular manufacture. And if you look at the reports, Janssen and other companies had lots of difficulty predicting the titers or predicting the volumes they could produce. And there are reports that some of the European manufacturing facilities, that the cell lines produce stubbornly low titers. So there's a lot of work kind of trying to work about process efficiencies in that area. And I think the most interesting one, and I think my colleague in the audience, Anne Moore, pointed this one out to me, is around the protein-based vaccines. And a key aspect of that is the adjuvants. And the adjuvant they use to stimulate the immune response is a matrix M. A matrix M has one component which comes from the soap bark tree in Chile. And every dose needs 1 gram of this bark to make a dose. So when the company were looking at that, they contacted their supplier and said, can you supply so many million doses? And he kind of goes, yes, kind of scaringly, but he couldn't, because that would be the whole soap bark harvest in Chile. It would basically decimate that. And to grow more trees, the sustainability of that, if that had been the one vaccine we were relying on, it would have been very challenging to produce sufficient supplies to manufacture. So it's a kind of an interesting area to highlight the scalability, how it can present in different areas. And I suppose anybody who's worked in pharmaceutical manufacturing at a manufacturing level will have come across their own stories. So what I'd like to do today is kind of go through some of the research we're doing and see how it ties in with scalability and the challenges related to that. And I'll just kick off with them. The first is this academic industry collaboration that I'm involved in, which is called the Manufacturing Classification System. So it involves a number of pharmaceutical scientists in industry and academia. And it's really looking at solid oral dosage forms and looking at the properties of the API and deciding which route to go. So you can go direct compression, wet granulation, dry granulation, or a more complex route. And what's really nice about this work and the two papers that have been developed in this area is they've actually looked at the API and they've looked at what do you need to consider if you're going to wet granulation. And I've just put up one of the tables there, but in the papers they've one for direct compression, dry granulation. And they've also, which is interesting, it's not a great graphic here, I don't know if you can see, it's not too clear, but they've also gone back and looked at a number of filings and company data. And what they've noted is that, okay, if you have a high drug loading and it's poorly water soluble, which is the top right side, the orange bar, then you're going to go to, companies are more likely to go to wet granulation, whereas when the dose is lower, it's much more spread. Now, there's a lot of factors that influence what a company will do and what their process, and certain companies prefer, it can be dictated by the equipment in the company's inventory as well. But they've kind of started to break down, and there's been a lot of interest from this from regulators, and also there's a new paper coming out where they're looking at how that's impacted who's citing this work and why they're citing it. And a lot of academic groups are using it now to determine if they're designing particles or granules, what do they need to bring it to the next stage, to the tablet manufacturer. The next project, which I'm going to spend a bit of time on, is a project that I was involved in. So it was, I was involved in it, it was the PMTC here in UL, and it was Alchemers. So this was funded through Enterprise Ireland, and it was really looking at, can we model formulation behavior at a commercial scale during blend characterization data, from blend characterization data. So when we start taking our API and we want to manufacture a simple thing like a tablet, and the simplest way is blend it with your excipients and compress it, then we can characterize this data, we can characterize this powder, and we can characterize it for flow, compaction, segregation, whether it segregates. So we can establish those properties. And then the next step is to put that on a R&D, a smallish scale tablet press, and to see how it tablets, how it manufactures. And then the next stage is kind of the tech transfer, where you're going now from that small scale press up to your bigger press models. And you're going to try and then conduct your DOEs or conduct your design space to see how it will perform on the large scale. And different presses will have different kind of operational processes. So here in the study, we looked at one R&D press and two different commercial presses. And really what happens at the commercial scale, you need to maintain your product quality, that's critical, but you need to maximize your production. So really what's key here, it's commercial, so you're trying to maximize your production. So you want those presses to go as fast as possible without undermining your product quality. And what we really want to do is to try and predict how the formulations would behave on the production scale presses for a new formulation. So if Alchemers had a new product coming through, they could characterize the blend and be able to predict what press to put it on and which press it would work fastest on. So that was kind of the challenge. And I suppose the project was envisaged by Alchemers, and a lot of I can't claim We came up with the plan, but we worked together on it. But a lot of the ideas around this project were from Stephen Sheehan and Claire in Alchemers. So what we did is, instead of looking at commercial products, what was developed with the help of Alchemers in the US were placebo formulations that would mimic the range of flow behavior of the commercial products that they made. So there's been a lot of studies done in this space, but they've tended to look at extremes, materials with extremes of flow. So these were good, medium, and poor flow, they were classified as. Then the next stage was each press. We put each formulation on each press and studied the effect of the press speed and the feeder speed. So we had 9 separate DOEs, and each DOE had, I think, around 16 runs. And we were trying to see, okay, on each press, how does feeder speed and press speed influence the weight variability? And from those 2 categorical, the formulation and the press, and the continuous variables, the press speed and the feeder speed, we developed a model, and then we validated that with a commercial data from a commercial product. And then we repeated the same on the compaction part of the tableting process with, again, placebo formulations, elastic, plastic, and brittle. They were the categorical variables. Then, again, we looked at this time on press speed and the compression forces. And again, we developed the model to predict hardness, friability, disintegration, and the design space around those. You'll note that I've put in coded because each press operated in different speed ranges. So our R&D press was quite slow, whereas our productions were fast. So you couldn't compare the speeds directly, but by normalizing that data, we could incorporate it into the model. And I suppose why we went with this approach of a DOE on each combination of press and formulation was because the reviewers came back and said, well, why didn't you go with a 5, why didn't you incorporate the press and the design in your factorial design? What it allows us to do, what it allows Alchemers to do is to look at what's presses in their inventory already. And then what happens if the R&D press breaks down and they have to buy a new one that operates a bit different? Well, then you can run the placebo formulations on that and incorporate that into the model, or if there's a new production press. And the same way, the model can expand to incorporate different formulation types. Okay, so what we developed really, it was a statistical model. It was a platform model that could predict commercial scale performance from small scale blend data. And then what the benefit for Alchemers is to streamline that tech transfer and scale up activities. And this just highlights some nice data that highlights the impact of it, was that if we look at this, if we have good flow, we can produce our Alchemers can produce under 3 million tablets per year on this module P press. If they've passable flow, it's less than half that. So this kind of data can really inform back to the formulators. If they say, oh, this is as good as we can get, well, it has serious implications if we go back here, it has serious implications if we decide on a formulation here that doesn't perform well. So it's kind of a very useful study from that point of view. And it's been published in 2 peer-reviewed publications. I think this touches on what Vivek was talking about a bit. This was a statistical study. It looks at causal links, and we'd really like to start moving up to more hybrid models where we can incorporate more mechanistic models and try and predict different parameters to identify the issues with the non-ideal behavior of real-world presses and real-world formulations. Okay, so am I doing okay with time? Okay, we'll go. So the next thing is really around the edge of failure. This is percolation work that Ana Luisa Cuerza did in our group. It's really when you start blending, often you increase the amount of drug, and what happens is there's a threshold on which you go over. So you can see here, the blue is the excipient and the white dots are the drug. So once we get to a certain point, initially the white dots are dispersed as islands in the blue, and we eventually get to a point where the blue dots are dispersed as islands in the white. You know, we start getting this percolation of drug particles. And what that can do is like an edge of failure. We can see here the weight variation. It starts to increase kind of between 15 and 20, we get a jump in weight variation. Our dissolution, we see the same behavior because now we've bigger lumps of drug. Even if we've done our nice crystals, they may be nice crystals and have better dissolution by themselves, but when you start compacting them into the tablet, we get this jump up. And again, when we were looking at dissolution at 5 minutes, we saw this again, a very dramatic change around 17%. So it's important to consider that as well when you're developing formulations. And just to finish off, what I want to talk about, mentioned scalability, but we also have scale up, we can have scale out, so continuous processing, duplicating rigs, and it's quite a common term in biologics manufacture for single-use manufacturing. And eventually, there's a lot of talk, it's probably very futuristic, but bedside manufacture, enabling precision and personalized medicine. And as we move, and I suppose this has been a topic, the group in Utrecht, in this Raymond Schiffler there, they're talking about it for RNA therapeutics, but there's also interest for orphan drugs, cancer treatments, to be able to manufacture at the bedside. And what I want to just point out is once you get to that kind of area, you're now increasing your variability and your process monitoring, and your quality assurance increases. And you have the same issues here, but what I think really is changing is we're moving, we used to be quality by testing, we went to quality by design. Moving to continuous now, we're moving to quality by control. But once we get to this precision medicine, we're actually back to quality by testing again, testing every product. And that's, I think, where the spectroscopy comes into play. Just showing some work from various students here. This was monitoring a co-crystal process using Raman, quite a common way. It's quite common to use spectroscopy to monitor processes. This is some nice work from Brian Kearns, who's just recent with collaboration with our Anne-Marie Healy in Trinity, using terahertz Raman to monitor the amount of crystalline or amorphous drug in a drug silica system, and some nice work by Khalid El-Kassas, working with Kappa in Munster Technological University, designing a fluorescence rig that can look at the protein stability in vial. So all these would support this idea of precision medicine and advanced manufacture. So formulation scientists, I think when I was a scientist, you just said, oh, is it safe? Is the material safe? And you go, it's GRAS listed. There's a whole discussion around that about what's safe and what's not safe. It depends on the route of delivery, but we need to move beyond that to consider other risks. That the APA properties can drive the processing route, but you can also design your properties, as we've seen earlier, to influence, to get the processing route you want. We need to, I'll just go through these quickly. We need to be aware of the edge of failure. It's lovely as an academic to show nice results, but it's really interesting as a scientist to see where the nice results stop and the bad results start. And scalability, we haven't shown it here, but it's a big area. Storage, humidity, environment, all that influences scalability. And scalability in the future is not just scale up, it's scale out. And so I think the rapid analytics, the PAT and the modeling, which we've heard so much about the last while, they're all really crucial that we can have scalable systems. There's loads of people to thank, I've put them there. And stealing from Jer's idea, we're hiring too. So we're looking for postdocs for industrial academic collaborative projects. Okay, thank you.